Assalamu alaikum everyone. Welcome back to Sn Academic. Today we are diving into one of the most groundbreaking advancements in medical science, that is liquid biopsy. It is basically a biochemical revolution in the cancer detection. So, cancer continues to be one of the biggest global health challenges of our time. But here's the truth: early detection can make all the difference. It can save lives and improve treatment outcomes dramatically. That's where liquid biopsy steps in. Unlike traditional biopsies that are invasive and time consuming, liquid biopsy is a non invasive, real time method that's changing the game in cancer detection and monitoring. It is also biochemically rich. Traditional biopsy is invasive. It means doctors have to cut into the body to to take a sample, and this can be obviously painful and risky for the patient. Also, tumors are not always the same throughout. For example, one part of a tumor might have different cells than another part. This is called tumor heterogeneity. You can see in this picture as well that as the time is progressing, the tumor heterogeneity is basically increasing. Different number of cells, different types of cells are increasing. So, but if we talk about the traditional biopsy, it only takes sample from just one spot, so it can miss some important information. That's why liquid biopsy is helpful. It uses blood or other bodily fluids to collect cancer signals from all over the body, and it makes it more safer and more accurate. Now, what is liquid biopsy? If we would keep all that information in mind, it is basically a test that can be performed on blood or bodily fluids to detect the cancer links molecules. What are those biomarkers? They can be ctDNA, that is circulating tumor DNA, CTCs, that can be circulating tumor cells, exosomes, or microRNAs. It basically offers a snapshot of the genetics of the tumor and also its biochemical activity, so it helps doctors understand how the cancer is behaving inside the body. Now, the principle of liquid biopsy is basically is that is uh, tumors release tiny pieces of material into the blood when their cells die. It can be either naturally called the apoptosis or due to the damage called the necrosis. They can also release things through secretions. These material includes DNA, it includes RNA, protein, or the small packets called the vesicles. So, liquid biopsy works by detecting and studying these tumor materials in the blood. Advanced lab techniques like PCR, digital droplet PCR, next generation sequencing, NGS, and immunoassays are used to find and basically analyze them. So, this helps doctor learn what kind of cancer is present and how it's changing, all without surgery. Now, let's talk about how liquid biopsy works. They are different steps. First step is the collection of the sample. The sample can be collected through by the blood or by any other body fluid. But the most frequently used and the convenient method is to collect the blood sample. The blood sample is collected in the same way a blood basically the blood sample that is collected for the CBC. It is collected in the same way. The blood is collected from the patient's arm, and it contains the tumor derived material like the circulating tumor DNA and circulating tumor cells. Also, the sample is also collected from the other body fluids, it depending on the tumor location. For example, if someone is suspecting cancer in bladder, kidney, or prostate, uh, then the sample would be collected from the urine, same then from the saliva or the cerebrospinal fluid or the pleural fluid. And also, one thing that should be kept in mind is that the samples are carefully handled. Why? To preserve the tumor material for testing, because they are liable to changes due to external conditions. The next step is testing the sample. After the sample is collected, basically the scientists would extract the tumor genetic material that includes DNA or RNA, and then construct a library. The constructing a library means organizing and amplifying it in a way that it becomes machine readable. Then the third step is sequencing. After the genetic after it, the genetic code is basically read by using the high-tech machines, and the mutations or the changes in the tumor's DNA or RNA is identified, and then the data is analyzed using bioinformatic tools, and this helps scientists to identify the type of the cancer's progression and whether or not the cancer is responding to treatments or not. So to repeat, we collect the sample, we extract the DNA and RNA, we construct a library, we then perform sequencing, and then we would analyze the data. Same, you can see in this picture as well that the blood sample is collected and then it is analyzed for all the biochemical markers that include circulating tumor cells and circulating tumor DNAs. Now, to compare the liquid biopsy with the traditional biopsy, for the liquid biopsy, as I have explained earlier, that the samples are collected using the samples from the blood or the other bodily fluids. While in tissue biopsy, that is an invasive procedure. The sample is collecting by surgically removing the tissue from a specific site, particularly from the tumor site or from the organ. So that is why liquid biopsy is called as minimally invasive, because it only involves a needle prick, while tissue biopsy is an invasive procedure, because in medical terms, those procedures that involves entering into the body cavity or those involving anesthesia is basically an invasive procedure. Liquid biopsy is suitable for how monitoring how the disease is progressing, or to detect that whether even after the treatment of the cancer is left in the body or not, or to assess whether the treatment we are giving to the patient is working or not. While the tissue biopsy is performed for the initial diagnosis, also to classify the tissues, and also the in-depth genomic profiling. Now, what are the target biomarkers that are analyzed using liquid biopsy? It includes circulating tumor cells, that are basically the whole cancer cells that have been detached from the tumor and that are circulating in the blood. Also, these cells would also release DNA, that is cell-free DNA or the cfDNA or the circulating tumor DNA. What it is? It is basically the fragments of DNA from the cells that comes specifically from the cancer cells. Other than this, circulating tumor cells also release small vesicles that carrying proteins, RNA, and DNA that are called exosomes and the extracellular vesicles. Other than this, there are microRNAs that are very small pieces of RNA that don't make proteins. Instead, they help control which genes are turned on or off by the body. In diseases like cancer, these microRNAs can change, and that's important. By studying them through liquid biopsy, doctors can learn more about the presence and type of cancer. Now, what are the types of cancer that are detected using liquid biopsy? These can be from the breast, these can be the lung cancer, prostate cancer, pancreatic cancer, colorectal cancer. It is especially helpful and useful in diagnosing or monitoring the progression of the cancer that is spreading, that is a metastatic cancer, or also those that are inoperable cancer, for which the traditional biopsy cannot be performed. For example, if someone is having a cancer in the brain, we cannot simply take a tissue out of the brain. It is risky and painful. So in that case, the liquid biopsy is performed. Now, what are the clinical applications? Biopsy has many important uses in cancer care. First, it helps in early diagnosis, finding cancer at an early stage. Second, it's used to monitor how well the treatment is working. Third, it can detect if cancer comes back or if any small amount is left in the body. And finally, it can help identify drug resistance, meaning the cancer stops responding to certain medicines. And all of this is done with just a simple test. Now, to conclude the discussion, liquid biopsy is a new, non invasive technique or minimally invasive technique that can detect disease biomarkers in what? In blood, in urine, in sputum. Other than this, the liquid biopsy is useful when not enough tissue sample is available, when not enough tumor tissue is in a sample, a tumor is hard to reach, like what I have explained in the case of the brain. And then when the regular monitoring is needed, and then why the liquid biopsies are basically used to analyze the presence of cancer cells, the DNA or the other substances that are released by the tumors, making It's very easy for us to diagnose or monitor the progress or to determine the effectiveness of the treatment. So that's all from today. If you like this video, then don't forget to like, subscribe, and share, and also comment down your questions below. I would love to answer it. Until then, Allah Hafiz, everyone.